Our next speaker is Mark Lawler, who is the chair of the Translational Cancer Genomics at Queen's University Belfast. And Mark is going to talk to us about the clinical cancer genomics projects going on.
Thanks very much, Heidi. John Byrne reminded me that I have to make my disclosure. This is my favorite disclosure from an Irishman. Just in relation to our committee, because this is very much a team effort, and then I'll focus on some individuals within the committee. I think the committee also shows you that really we are global in our nature, and that obviously brings advantages. It also brings disadvantages. So it means that Catherine North is just making her coffee for breakfast the following morning while I'm just waiting for the cold to end so I can have a glass of Sauvignon Blanc. Peter Goodhand suggested I was probably having the glass of Sauvignon Blanc at 8 o'clock in the morning, but that's not true. Okay, so what I want to do really is just tell you a little bit about, sort of where we think we're going. Obviously, we're an emerging project, highlighting some of the things we've done so far, and also at the end, just talk a little bit about some of the things that actually have developed during this meeting that I think show you why the Global Alliance works. What I want to do is just highlight really, and it's words that have been used frequently, both today and yesterday: compatibility, accessibility, scalability, but most importantly, sharing and linking. And because really what we're trying to do is identify potential needs, but then also provide solutions. And David, when he talked at the start today, highlighted, we need now to be in that implementation phase. We've danced with each other a lot, we've got to know each other. We now need to move that forward in relation to really delivering. Because at the end of the day, as Peter has said, it's really for the patients that we are here, so we really need to start delivering. Obviously, there are demonstration projects that started within the clinical working group and within the clinical cancer task team, which have now gone on to really show you, and I think John gave a brilliant description of really, and Heidi also, of two projects really that are showing real benefit and really are moving forward in relation to BRCA and Matchmaker. I want to talk particularly about cancer. Charles and myself co-chair the clinical genome task team, and really that's what I'm going to focus on today. What we really want to do is look at highlighting what's going on in the community, but then looking to see how GA4GH can add value really to those initiatives and how we can really bring together a lot of the initiatives and also potentially set standards, best practices moving forward. We have, over the last few months, have really worked quite hard together to produce an opinion paper, which we're shortly going to prepare for publication, or for submission, I should say, and that will really highlight to the cancer community and also the overall community, genetics community, what's happening within this space. And we know, and we've heard in relation to the whole, what we're trying to achieve really, but just to highlight it, just to emphasize it again, big data genomics is not going to be effective the way we are currently doing it because of these reasons. Short-sightedness in relation to looking at smaller data sets, lack of interoperability, you've heard David Hassler talk about that a lot, but also this reluctance to share. And really we have to change that mindset. And I think you've seen over the last year that that mindset is really changing, and some quite big initiatives are partnering with the Global Alliance in terms of how we actually can work together and give added benefit, because I think that's very important. And that added benefit, which allows you, for example, from the biology side to get increased granularity, but also that increased granularity can inform clinical benefit. The second thing, and I do want to highlight this a little bit, and because I'm actually reaching out to you on part of myself, Lillian Sue and Emile Vost, is we are actually looking at now developing a survey. This survey is nearly ready for sending out to the community, and we really want you to participate as members of GA4GH and to spread the word in relation to this survey. What we want to do is really highlight and identify ongoing data sharing efforts in the cancer space, and that will really allow us then to bring all those groups together, to work together, to answer some of the key questions that we want to in cancer. And so we already know no single hospital, no single institution can go it alone. And our problem, particularly in cancer, is we might start with quite large data sets, but when we parse them down in relation to molecular taxonomy, for example, the numbers of patient samples, the numbers of data points that we actually achieve in individual data sets can tend to be quite small and can't really answer some of the great challenges in cancer that we're faced with. We want to emphasize the need for responsible data sharing, uniformity in the collection of data, but also that we're speaking the same language at the IT level in relation to different bioinformatics platforms. So we feel that doing this will allow us then the potential to create a better environment that really will increase, for one example, increase either both new but also repurposed therapies in the context of both clinical trials, but also in the context of real-time data that's generated with our patients. So this will be a survey, followed then by a white paper. It'll delineate the importance and the benefits of data sharing, highlight examples ongoing or planned throughout the world, discuss key challenges, and these challenges are significant. You've heard from a number of the speakers this morning that there are not, by no means, challenges that we can face easily. But as a community, as we work together, we've really shown, I think, the ability to step up to the plate and actually try and hit that home run. And also, we really want to emphasize, I'm going to emphasize several times during the presentation, the importance of implementation. We have to both talk the talk, but also, or walk the walk, but also talk the talk. This is just showing you some of the details of the survey. I'm not going to go through it in detail, but just to highlight some of the elements we're going to be capturing from the community itself. So that's why I'm reaching out to you really here today to ask you to participate in this survey. And my email will be at the end, but also I'm available at lunchtime really to talk over it if people are interested in participating. And this just shows you our timeline. So we're aiming to launch the survey this month and then start writing a manuscript, a white paper associated with the results that we get from the survey, analyze the survey, and then submit a manuscript towards the end of September. The third initiative that I want to spend most of the remaining time on is a brand new initiative, and it's really still in its nascent phase, so to speak, and that's the Actionable Cancer Genome Initiative, which is led by Charles Sawyers, and you can see collaborating with a number of different initiatives, ClinGen, American Society for Medical Genetics, and American College for Medical Genetics and Genomics, Cancer Core Europe, Genie, EORTC, etc. The origins of this, Rick Klausner, former director of the NCI but chief medical officer at Illumina, really brought together different leaders from the cancer centres that I've indicated there and looked at developing what was called an actionable cancer or an actionable genome consortium. Series of telecoms, face-to-faces, and the product of this, Charles Sawyers, is driving the writing of an opinion piece, which will be ready shortly as a white paper. And this really represents the best practices by thought leaders within clinical somatic sequencing. What we feel is that there's a potential for GA4GH to foster this initiative, nurture this initiative, drive it going forward at a global level. Just to let you know, obviously it has some similarities and overlap with ClinGen. ClinGen, the Clinical Genome Resource, and you heard Heidi talk about this in detail yesterday. And just highlighting that this program is a very important program, and we see this as a complementarity in relation to how we move forward and work together in relation to the Actionable Cancer Genome Initiative. Just again showing you the importance of ClinGen in relation to looking at clinical validity, but also clinical utility. So how best can these different groupings work together, and how can GA4GH help that process? As I said, we're proposing the creation of an Actionable Cancer Genome Initiative, and really the benefit, and Warren Kibbe would have emphasized this yesterday as well, one benefit, for example, in matching clinical and genomic data with response data. And really using that information, it's a space that we need to address and would be empowered by a joint approach. And then bringing the international community together to actually participate in this initiative. And again, you would have heard yesterday from Cancer Core Europe and from Genie in this regard, and Fabian is here, and I'm sure he can contribute in the questions. And so I'm setting a challenge, or we're setting a challenge to you. What do we need to really get a somatic cancer genome clinical annotation effort to the appropriate scale that will serve as a resource for the community? So that's a challenge I'm putting out there to you. We want you to respond. You've heard from other speakers. We want you to be part of this initiative with us and working forward with us. So in terms of going away from this meeting, we want you to think about this and really come back to us and respond to us. The potential goals of such an initiative, really to look at the validity of genes to be tested on NGS platforms, perhaps establishing an actionable cancer genome panel. Does it really make sense to have 20, 30, 40 different panels and testing them, producing small amounts of data and then finding that they're not interoperable? I don't think so. Also looking at optimal standards in relation to the technical aspects of NGS, looking at both curation and annotation approaches, really because we're seeing an exponential increase in the number of variants in the somatic space in cancer. And then two other things, and I think it's something maybe we haven't talked about a lot so far today, but I do highlight them as being important areas. One is facilitating pathways to regulatory approval and reimbursement. And then one of the things we've come up with is this idea of flexible, clear principles that allow the test selection to be usable, billable, payable, and sustainable. Because otherwise, if the moment we're relying a lot in relation to individual institutions being able to do the NGS at the scale that we're talking about, really, but not in a way in which it's reimbursable. And that's not sustainable over the long term. So really, I think GA4GH, or we think GA4GH, really needs to step into that space and be authoritative in relation to this. Two initiatives you heard about yesterday, Genie, and looking at 8 different institutions, 6 in the United States, 1 in Canada, 1 in Europe, looking at approximately 100,000 patients within 4 to 5 years. A second initiative, Cancer Core Europe, again, 6 different institutions throughout Europe sharing a common IT system, already have 4,000 genomes in that context, and the ability to start to analyze those and looking at bringing that community together. So what are the type of things we need to be thinking about, though, if we actually sort of drill down a little bit further in relation to the actual cancer genome? One of the things we realized, the importance of genetic context. We realized it quite quickly in relation to EGFR therapies in cancer, where we actually really needed to have the context in relation to KRAS mutational status. We know from the excellent results, superb results that were just reported recently in ASCO in relation to targeting BRAF mutations, that it really works in melanoma. However, it doesn't work at all in colorectal cancer. So aren't they the type of questions that if we had a federated approach that actually brought the different initiatives together, that we could really start to drill down and get that level of granularity that allowed us to ask these type of questions and provide solutions that then would drive particular therapeutic approaches for patients who don't respond to the targeted approach with BRAF. So the other thing I would just emphasize is we talk a lot about reaching out, collaborating, sharing, but we also need to practice what we preach internally. And one of the great things about yesterday, for those of you who were here, was actually that we had a joint session between the clinical working group and the data working group, so that we could actually work together. So rather than having separate streams, we actually work together. And out of that, and out of the discussions in the afternoon in the 2 of the sessions, we've now set up, for example, a cross-cutting cancer driver project planning group, chaired by Adam Margolin from Data Working and myself. And we can start to look at the challenges in relation to genotype to phenotype. Do we want to ask a very simple question? The question we asked in relation to BRCA challenge was a relatively simple question. Do we want to ask a similar question in relation to the mutation that we see in melanoma? One mutation, if we look at that in the context of different cancers and really ask, why do they respond in some situations, why not in others? Can we get that level of data in relation to genetic context that really helps us to drive targeted combinatorial approaches? Immunotherapy. Anybody of you who was here was at ASCO, immunotherapy was all over the place in relation to, it was not, I don't mean all over the place in a negative way, I mean it was featuring heavily in all aspects of the program, and I'm sure Peter can comment in relation to that. So if we could actually say, well, how do we actually predict which patients will respond to anti-PD-L1 therapy, for example, that would be another approach that we could think about. But most importantly, we need to, and this is why the cross-cutting group has been set up, we really need to develop our APIs all the way up, but use real data to actually test them And that then allows us then to test them in one particular situation, but then to make them applicable across a wide range of situations. And again, just emphasizing once again the importance we need to implement. So let's overcome the silo mentality that was present before, but I think we all agree is starting to change. We don't want to be islands in a data stream. I'd like to acknowledge our executive committee, particularly the leadership of both Charles and Catherine, which has been inspirational, the clinical genome task team, Lillian, Sue and Emile in relation to the survey that I mentioned, ClinGen, particularly Heidi, the American College of Medical Genetics and Genomics, and Mike is here in the audience as well, and obviously Rachel in terms of her super support for our, both at the executive committee level and the task team level. And I'm happy to take any questions. Thank you. Do
we have any questions? So maybe I'll get us started. You know, this is an incredibly complex area with a lot of people involved. And what do you think is Global Alliance's sort of first step in how to help orchestrate collaboration here? Is it just simply bringing groups together and figuring out who's doing what? Is it tackling a small project as a demonstration? How do you see the best way for us to start out?
Thanks very much, Heidi. I think two things. I think one, the survey that we're doing is going to be very important because not only does it give us information, it also brings linkages. So it does two things. It gives us the information we want, but it also actually provides those linkages to bring people together, because obviously in relation to a number of tasks that we might want to do, we're going to need different expertise, and also we're going to need that, you know, sort of learned experience from each other. In relation to the second aspect then is really, that's where our cross-cutting team comes in to identify, and I think, you know, we've said it before, Catherine has said, bite-sized chunks. Don't try and do too many things. Try and do too many things, you fail. It's no use being excellent at nothing. Do one thing really well. And so that's really what we're going to be discussing within that group and identifying one project or driver project that we can move forward. There are obviously things that we can link into that are already happening in relation to Beacon, Matchmaker, etc. But I think in terms of that, we don't want to oversell ourselves in relation to what we can do.
Hi, Professor. I have a question. In my understanding, I don't know whether it is correct, because when I listening about the project like a Matchmaker project and the BRCA challenge, this kind of project, in fact, the data, when we talk about data sharing, in fact, it's like a data as global commons, right? Because everyone can use this data, but I don't know that in this project whether it is the same thing, because this is clinical data, and when you talk about like some project, it is like a cooperation between several institutions themselves, that they can share the data with themselves. But so it sounds like this data sharing is a little bit different from the other data sharing we mentioned today.
I mean, there are differences, but there are also complementarities. I mean, I think the two programs we mentioned, Genie and Cancer Core Europe, there are obviously others as well. One of the conversations we've had is linking those two projects together. They obviously have commonalities in relation to Gustav Rossi being involved in both. The Cancer Core Europe is a very good example. Fabian might want to comment on it, because theirs is a common IT sharing program between the 6 centres. Yes, clinical data sharing is difficult, but I think that's why we want to work together with the data working group in terms of finding what are the solutions that we can develop. And that's really, if I've got one thing out of this meeting, it's a meeting that happened yesterday afternoon where 10 of us sat down after one of the sessions and actually worked together and said, okay, what are we going to do? How are we going to do it? How are we going to move forward?
David?
So one of the things you raised, and I'd like to ask you to comment more about, is regulators. So as we've talked about, what I find myself thinking through all the talks is, how do we actually drive change? What will actually drive vendors and users? And one answer is regulators, right? Because research is not very heavily regulated. So if the researchers in the room, they may not be as aware of how to do anything in the clinical space or as a company in the diagnostic space. There's often a lot of regulation. In the cancer genome space, where there's so much move towards targeted therapy and basket trials and the like, what are the regulators looking for? And is there some way that we should be engaging with them as a way to drive uptake, is to try and interact with them?
David, I think we have to interact with them. I mean, I sit on another body called the European Alliance for Personalised Medicine, and we're looking at exactly that issue. And we organise, for example, sessions with the regulators, also sessions with the European Parliament in relation to actually looking at what are the best ways. Because I think otherwise we're not going to get to that implementation phase. We're going to have a lot of nice things that we've done together. We're going to write nice papers together. We're going to do all that part of it, which is an important part, don't get me wrong, but we're not going to take that next step. So I do think we need to sort of think with our sort of policy hats on in relation to how we can do that.
Yes, question?
It's more sort of a comment to the Global Alliance. Ernst Hoffmann from ETH Zurich.
Hi, Ernst.
Hello. And so we heard many times of how data integration, getting data out of silos is important. And we also heard in the previous talk that the human individual is actually the integrator of all the data. And I think we should actually use this and empower the individual to contribute. First of all, I didn't know this until 2 weeks ago, there is a human right to science. And it explicitly states that individuals, lay people, should be allowed to participate in scientific projects. Now you have 2 million people on Zooniverse participating in classifying galaxies. Can you imagine how many people you will have to participate in scientific research in medical that affects themselves? So our initiative for the past 3 years is actually to provide my data cooperative databases where each individual can open an account and put all his medical and e-health and genome data in and share it with whoever he wants, doctors for second opinions or research. And I think if we engage directly the individuals and empower them by having this cooperative organization that is owned by the citizens and not by some shareholders, we can regain the value of the personal data economy, and we can democratize it. And for health, we can advance by engaging with the citizens as partners in these citizen science projects.
Absolutely. I mean, you highlighted the whole area of citizen science, which I think is something that we do need to actually address. Certainly, it would have come up when we actually had at the European Society of Human Genetics meeting in Glasgow just there on Sunday, in relation to the round table that we had, or the panel discussion. We actually talked about that quite a lot. And Anna Middleton, who's a social scientist who works at the Wellcome Trust, had some very interesting ideas, including this idea of socializing the genome, which I think is something that really is very interesting, has a lot of, and I know we have a group that is starting in relation to access. So certainly I think, if you look in the States, Bob Nussbaum has work in relation to BRCA, in relation to actually, patients driving the data. Heidi highlighted it as well in relation to both in her own talk and also in questions. So yes, I do think it's something that we need to address and use working forward in partnership.
Okay, well, if we're out of questions, I think we are ready for lunch. Any instructions, Peter?
I was just going to ask that. So the earlier question about the sharing of data. That earlier question about the sharing of data between specific institutions, but my understanding was that the outcomes, the learnings from that would be broadly available.
Yes.
So that actionable panel is not just to be kept within those.
Not at all. No,
and global knowledge coming from selective
not
sharing.
at all. Sorry, if I gave that impression, that wasn't the case at all, no. Just as well, if anybody's interested in relation to the survey, either email me or I'm here at lunchtime, you can come and chat to me. Thank you.
The only instruction I'd have for lunch is it's going to get crowded, so if you're really, really hungry, rush. If you're not, do your email, go for a walk, and wait till the queue dissipates. I'm not sure. What time who's got after, what? 45? So back at